Good day everyone. I'm Nino Jaime Troceno, second year student of Bachelor of Science in Agriculture. So I am here to discuss the environmental influence of gene expression. So first is the external environment. External environment is defined as the effects are those leading to phenotypic changes that seem directly correlated with observable changes outside the organism. So, Sylvie, this part is observable ra siya or makita ra siya, visible in the eyes. Or ma-feel ra nimo siya. There are four of it. First is the temperature, second is light, third is nutrition, fourth is maternal relations. So, temperature, there is a close correlation between the rate of chemical reactions and temperature. Example, red color of the flower primrose at room temperature and its white color at temperature over 30 degrees Celsius. So about this, temperature has involved the result of changing the colors of the flower. So light energy provided by light, and light itself is essential for the growth and development of plants and animals. Example, seedlings grown in the dark do not survive long because chlorophyll is not developed even though there are genes for chlorophyll development. Third, nutrition. Food provides energy for carrying out necessary processes and materials that must be incorporated into necessary structures. Genetically, different organisms, even of the same species, usually differ in the kind of amount of nutrients they require. Example, the appearance of yellow fat in rabbits depends on the genotype YY and green vegetable eaten. So this depends on the nutrients they get every vegetable that they ate. Fourth is the maternal relations. In case of mammals, where the direct body relations between parent and progeny extend beyond fertilization and the interaction between the fetus and the material environment occurs. Example, blood group incompatibilities between the mother and offspring produce special effects of the survival of particular genotypes. So let's proceed to the internal environment. Internal environment is defined as the effects are those leading to phenotypic changes that appear correlated primarily to changes within the organism. So this happens within the organism or inside the organism. First one is the age. An individual grows older, there are certain phenotypic changes that occur which allows further genotypic effects to be expressed. So this is visible to us. When we age, some parts of our bodies will grow. Second is the sex. Sexual phenotypic differences are evident in the reproductive structures and specialized. Third is the substrates. The kinds of reaction that take place in an organism depend largely on the substrate present. These substances are synthesized by the organism and their presence or absence may be controlled. So now let's proceed to the chemical basis of heredity. DNA is known as the chemical basis of heredity. DNA is made up of combination of nitrogenous bases, sugar molecules, and phosphate groups which are linked by different bonds in a series of sequences. The DNA is responsible for the genetic makeup of an individual. It has the important role of carrying and transmitting the hereditary materials from parents to offspring. The transfer of genetic material starts with the reproduction. During fertilization, the sperm from the male and the egg from the female used to form zygote. The genetic material, the DNA from both the parents, is combined in this process. Through cell division, meiosis, and mitosis, the DNA is split many times and transferred to the offspring. The new offspring inherits exactly half the DNA from each of its parents. The genetic material has greater variation among the parents itself, so that the combination of genetic material is different during each fertilization process. This brings variation among siblings also. The mix is unique. The mix of DNA is unique to each individual, as only 50% of the DNA is received from each of the parents, who receive 50% of their DNA to their parents, and so on. Hence, the DNA keeps reducing over generations. The DNA also does transcription and translation of proteins inside the cell. The information to produce a particular protein is encoded in the DNA. The DNA undergoes transcription to produce the messenger RNA, in which the information about proteins is copied. The messenger RNA is then transferred to the ribosome, where it reads the information and starts assembling the amino acids into a protein. It is known that the proteins are one of the most important organic molecules in living systems and have a diverse range of functions inside the body.